Hello, thank you for taking the time out of your day to listen to this presentation here as part of the Antibody Engineering and Therapeutics Conference, of course being held virtually this year. And what I'd like to do today is give you an example of two case studies where we at ATOM were able to leverage some of our technologies to be a part of the pandemic response to the current SARS-CoV-2/COVID-19 situation we all find ourselves in. Before I do, I'd like to remind you a little bit about who we are. Originally founded as DNA 2.0 back in 2003, nearly 18 years ago. We are an independent organization, about 120 people, and we're a CRO, but we're very heavily focused on innovation and on the science, as demonstrated here by the number of patents and papers that we contribute to. And of course, we're located in the San Francisco Bay Area. Much of what ATOM has built over the years is something we refer to as the GPS platform. This is a platform or a methodology by which certain attributes can be optimized inside different biological systems. Could be applied to non-biological systems, but here in biotech, that's of course what we focus on. And it begins with the design of experiment. This happens in silico, where you use different permutations and data sets to design in silico different variants of something that you would like to test. Of course, those need to be built at some point out of the silico and in real life, tested by virtue of whatever biological or expression or physical tests need to be performed. And then, of course, we use machine learning to try to model the data set to understand what attributes of these different variants created what outcomes in the final model. And of course, this is an iterative process, and it goes around and round, and as the model gets better and better, we get to a situation where the platform is able to predict something with relatively good certainty. Now, as an example, you could think of this as codon optimization, where here what you've done is coded the same gene with different sets of codons. You test for expression, you try to model that expression and go around in a circle until you come up with an algorithm that then is able to do codon optimization. In fact, codon optimization is one of the key pillars of our GPS platform. We call it Gene GPS. In addition, we have a vector GPS platform where we vary the vector elements to optimize expression or to alter it in some way. And of course, we now also do protein GPS, where we're engineering proteins to have very particular attributes or activities. And it is in fact the outcome of one of these projects internally where we actually created something called the leap-in transposase. This is a transposase enzyme heavily engineered for moving DNA into genomes. And it's part of the core part of the transposase platform, which of course includes also the gene GPS and vector GPS elements. And I'll come back to the leap-in transposase system towards the end. But the first case that I'm going to talk about is actually not with respect to the leap-in transposase. This is a client of ours. As such, I can't show you very much of the data, unfortunately, for confidentiality reasons. But suffice it to say, they have a very high throughput antibody discovery platform that they turned on immediately when the pandemic started and started identifying thousands and thousands of antibodies for both therapeutic as well as diagnostic applications. In this case, they came to us because they had triaged this down to 65 unique antibodies for a diagnostic application, and they needed immediate access to at least 30 milligrams of 57 of these antibodies, which we chose to run at 100 ml scale. And they had some higher priority antibodies, 8 of them, that they wanted at least 100 milligrams, and we chose to run this transient transfection at a 500 ml scale. And our solution to this begins with a fully integrated LIMS-driven process. We have a custom-built software solution which allows us to track from electronic sequence, whether it's an email or an upload into our system, all the way through to the shipment of protein with exquisite detail, with timestamps and data stamps for every single part of the process, which of course starts with gene synthesis, vector construction, and an optimized 2-vector platform, one for the heavy chain, one for the light chain. This then needs to be handed over to the transient expression group, which ran a 7-day fed batch process in HEK, and that needed to be handed off to the protein group, of course, which did a single pass protein A purification, followed by formulation, back to project management for shipping. So these are the steps in the process outlined here, and as you're no doubt aware, this tends to be a relatively long process. It goes from one group to the next, and there's handoffs and there's communication, and typically requires Some of it may even be outsourced and typically requiring on the order of 6 to 8 weeks. And now imagine if this group was all of a sudden asked to do this for 65 antibodies in parallel under pandemic response timelines. Needless to say, this whole process as it exists today is way too slow to be useful for that. So what was our timeline like, and were we able to deliver? So I've already mentioned we do all of this in-house and fully integrated, and so the team knew at all moments where each antibody was in the process, whether it was breezing through with no problems, whether there were some gene synthesis issues, whether it was in plasmid production for transfection, whether it was been transfected, etc. And so as a result, we were able to go from electronic sequence to purified, formulated and shipped antibody in just 18 days. Clearly just a fraction of the 50 to 60 days that's normally required to do this. You'll notice the asterisks here, starting with the transfection process. We did run into a bottleneck starting here in the process timeline, and so we decided to tranche this part into 3 tranches, resulting in 3 different sets of shipments. And so the final shipment was done just 24 days from the beginning. Again, a single pass was 18 days, and all 65 antibodies were delivered in 24 days. So the timeline was spot on. We were able to get this out to the client, but the question was, did we give them any protein? Did we give them sufficient protein for their needs? I mentioned we ran this at 2 scales, both the 100 ml scale and the 500 ml scale, and this platform is completely scalable, at least as we show it here. We actually scale it all the way down to 2 mils if we need to, but in this case, the yield at both 500 and 100 milliliter scale was in excess of 0.5 gram per liter. And to be clear, this is the formulated yield that was shipped. This is not the titer and the supernatant. This is not just past protein A. This is post-formulation in the vial shipping a volumetric yield of over 500 milligrams per liter of culture in both 500 and 100 ml scale. And of course, there's a distribution to that. And that ranged from about 250 to nearly 900 mg per liter, again, in purified yield. And you can see what you would expect with some antibodies expressing and behaving better than others. And how did this result with respect to the actual milligrams required? At the smaller scale, we were looking for 30 mg. We did, in fact, have 4 antibodies that were under 30 milligrams. You can see them down here. 3 of them in the 23 to 25 mg range. They didn't quite make it. This 4th one here has 29 milligrams, so technically it did not make 30 milligrams. But needless to say, the vast majority of the rest of them were well in excess of 30 milligrams and sufficient for the needs of our client. This is the 100 ml delivery. Here's the 500 ml, self-explanatory. All of them over 100 mg, ranging from 160 to 387 milligrams. So as you can see, we were very fast, high titer, robustly, and again, very exciting to say, this resulted in a program where we went from 65 antibodies in an electronic sequence to delivering protein in just 24 days. As I mentioned, this is a very high throughput group. They discover thousands and tens of thousands of antibodies on a routine basis. And this was a comment that came from one of their team members where they said, and I quote, "We've never seen delivery like this. Typically, our partners cannot keep up with us, but now we have to try to keep up with you." So that was very reassuring feedback from our client. Now I'm going to switch over to the second case study. In this case, we're going to therapeutics. Again, it's a client project, all this data under confidentiality, so I can't show you very much, which I apologize for, but I think the message comes clear. They had 11 candidate therapeutic antibodies, and obviously, as we all do, had a very strong desire to try to get this into human trials as quickly as possible. To do this, we had to help them focus on that sequence to phase 1 part, and specifically, of course, can we create a cell line substrate that can be used for manufacturing that is going to be that can be created quick enough, but also be robust enough to be compatible with a phase 1 process? And I mentioned the Leap-In platform. I'll come back to that again in a minute, but suffice it to say, we were focusing on the generation of cell pools, not clonal cells, for this manufacturing process. Since we didn't know how these antibodies would behave, we decided to create 2 different expression vectors for each one. To be clear, each vector has both heavy and light chain on them, but they have different regulatory elements. And then we created Leap-In transposase-derived pools for each one of these. Again, this is 11 antibodies, 2 vectors, and as such, 22 pools. The goal was to scale these up as quickly as we could, freeze down appropriately sized banks for transfer to the CDMO for manufacturing purposes, and in parallel to try to get some idea. Again, we're moving very, very quickly here. How quickly can we get some notion of what the expression level and the titers and how the protein looks coming out of these pools? And to do that, we implemented a 10 milliliter tube spin format. Not a shake flask, not a bioreactor, just a small tube spin, so 10 mils. Standard cell line development is, of course, a very long and tedious process. It takes 6, sometimes 8 months. It can be faster. We fully recognize that, and obviously way too long for what we're talking about. And that's why we were looking with this client to understand how we could best utilize pools. In a standard cell line development workflow, using non-targeted integration, non-leap-in integration, these are random integration events typically. This is a 3 to 4 month process that results in relatively low titer pools that are also quite unstable. If you've got to scale them up to 200, 500, or maybe 1,000 liter, the chance of you having a robust titer at that stage is quite low. And what this means is that using standard technologies, there's quite a high risk to trying to create a pandemic response phase 1 therapeutic using random or more traditional technologies. So it's way too slow, of course, and way too risky. You could go down this path and not get the protein out that you need. So the leap-in platform, I've talked a little bit about it. I'll talk about our standard timelines, which are already quite rapid. It's about 12 weeks from the transfection event through to the research cell bank, so it's already quite quick. And we were interested, of course, not so much in clonal research cell banks, but how can we get to the pools? Now, one of the key pieces of the transposase technology, the leap-in platform that I won't have time to talk too much about, is that our pools are actually very representative of the derivative clones. As such, our pools are actually quite high titer, they're quite stable, they're quite predictive of the future clones, and in the aggregate represent a relative low risk of taking these into phase 1 manufacturing. You're going to have the titer, you'll be stable enough, and if you have the opportunity to go to phase 2 and beyond, it's relatively low risk in terms of transitioning a process to the clones. But the question is, that's great, but how fast can we really go? I've already talked about this process in the previous case study. In this particular case study, we were approximately 7 days to go from the electronic sequence to transfection-ready plasmid. And again, this is in a process that will see regulatory scrutiny at the end of the day. So that took us about a week. And the question was, how long is it going to take to transfect, select out robust pools, scale them up, bank them, do a fed batch, and get them over into the CDMO's hands? So I mentioned we had 2 different vector sets. So let's look at how rapidly we were able to select the pools. The first vector is kind of our insurance policy, if you will. This is a vector we use for many projects in the past, and we understand well how it behaves. And what you can see here is selection in about 7 to 9 days. We get dips, not much lower, about 75% viability at the worst, recovery to over 90% viability in just 7 to 9 days. And you can see here robust passaging after that time as we're scaling up for banking and for our fed batch. This is the first vector set. The second vector set I may have mentioned. We had a good sense that this one could be even faster, and in fact it was. We had some data that suggested it would be true, and this is the first time we were able to do a quite extensive comparison, 11 antibodies, 2 different vectors, and here you see selection in just 3 to 5 days back to 90% plus viability. And again, robust passaging after this as we scaled up. So what you can see here is extremely rapid and robust selection of these pools. So you're starting to get a sense now of the timelines that we're talking about. 7 days to go from sequence into the cells, another, let's call it 3 to 7, maybe 10 days at most to get your pools out, and then it's just go to the races and see how much bulk biomass we can get to bank, to do a fed batch, and get this into the CDMO's hands. That's all from the timing perspective, but again, you've got to have enough protein to make a difference. So what do the titers look like? Here are the 11 antibodies. Again, not surprising. They have different titers when they're going through this process, but nonetheless, selected in 7 to 9 days, put into a small 10 ml tube spin format. Not a bioreactor, not an amber system, not a shake flask, just a tube spin. And we're seeing between 1.5 and roughly 3.5 gram per liter titers at day 14, quite respectable for such a rapid process, let alone even a more standard process. And vector number 2 did even better. Not only did they select better, but here we're seeing titers in the 3.5 to nearly 5 gram per liter range. Again, to be clear, these are cells that have been selected up to 90% viability in 3 to 5 days, demonstrating up to nearly 5 grams per liter in a small scale down tube spin format. So we're fast, we've got high titer, and then we banked these and sent them out to the CDMO. They're scaling them up for GMP and doing some stability data in their hands. I don't have that data. So what did this look like? How did this accelerated timeline come to bear as we leverage the leap-in transposase platform? I've talked about the gene synthesis up through the plasmid prep. We're talking about a week here. Pool selection in a handful of days. And a seed train, scale up, bank the pools, and then get them tested before they go to the CDMO. In parallel with the banking, I mentioned we were just creating biomass to do this fed batch run. We did the fed batch run once we were confident we were going to hit the timelines. Again, 10 mil tube spin, and then we shipped to the CDMO once we had the data in parallel with the testing so that they could hold these banks in quarantine at the CDMO. So what was the overall timeline from uploading an electronic sequence, shipping to the CDMO, typically something that takes many, many months, let alone if you're going to do it in pools, we're talking about 4 or 5, maybe 6 months. This was completed in 38 days. To be clear, electronic sequence, shipment of banks to a CDMO in 38 days. And how did the cells perform? They're performing beautifully in their hands. They're stable enough to scale them up for the needed volumes. They've done multiple GMP runs now. The IND is pending. We're hoping this year, here in December, before the new year, we'll get this IND. And more impressively than the time, or equally as impressive to the timelines we're talking about here, these cells in an intensified fed batch, talking 100, 200 cells per mil, we're looking in excess of 12 gram per liters. We're hearing rumors it could be as high as 15 or 16 grams per liter. Nonetheless, again, cell pools on an extremely rapid timeline moved into a novel and innovative intensified fed batch process, robustly generating 12, 14, maybe 15 gram per liter. And you can imagine for a pandemic response, that's going to create a lot of protein. We're talking about kilos per 100 liter reaction to treat the patients. So very simple take-home message here. Again, we're talking about therapeutic candidates from email to CDMO in 38 days. So with that, I'm going to go ahead and stop and thank you for your time. I've given you 2 case studies. Hopefully this is coming through in the virtual format, how exciting this is to us to be a part of this response, to see the GPS platform and the leap-in transposase to be able to provide value, not only for these 2 case studies which I described today, but many, many, many more. We're working on vaccines as well and creating cell lines for manufacturing there. We've done a lot of work to express not only the full-length S protein, but also different variants of the receptor binding domain for different applications, vaccines and diagnostics. And suffice it to say, have synthesized and created thousands of genes and gene elements for R&D, for academics, for large pharma, for many of our thousands of customers out there. We thank you for the opportunity to work with you as well. So with that, I'll go ahead and say thank you once again for your time. I really do appreciate it. Again, hopefully this is coming through in the virtual format, and I look forward to your questions and to seeing you all next year. Take care.